I'd like to begin today's presentation by telling you a joke. I really can't take credit for it. My wife made up this joke, and the only reason that she did it is because my daughter was, for some reason, wanting us to teach her new jokes and to come up with our own original material. My daughter's 5 years old. Anyway, I may have told you this joke before in class, but dang it, I don't care. I'm going to tell it to you again because I think you could use the entertainment. So here goes. It's a knock-knock joke, and since there's not really any interaction through these videos, I'm going to have to do both sides. Knock-knock. Who's there? Grape. Grape who? Grape who? You should go see a doctor. Ha 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 ha. Now let's get back to business. Practically all organic reactions in living systems require catalysts. Most biological catalysts are enzymes, which can increase the reaction rate of some reactions by a factor as high as 10 quadrillion. The reactant of an enzyme-catalyzed reaction is called a substrate. Just so you know, most enzymes' names end with the suffix ase. I only mention that because in the coming weeks you'll start to hear me say ACE so many times it'll drive you crazy. Invertase, protease, rediculase. These are all the names of enzymes. Now, enzymes bind their substrates in special pockets called active sites. In this computer-generated model, we can see an enzyme binding its substrate in its active site, much like a dinosaur eating a cupcake. Enzymes are usually comprised of multiple polymers of hundreds or even thousands of amino acids, and they're usually very specific for the substrates they bind. In fact, some enzymes won't tolerate even the slightest structural variation in their substrates, while other enzymes are a little bit more versatile. So how do enzymes bind their substrates? Well, back in 1894, Emil Fischer proposed a model called the lock and key model, depicted by this depiction here, in which he postulated that enzymes' active sites have a shape that's perfectly fit for the substrate they bind. Later in 1958, Daniel Koshland proposed a more generally correct model called the induced fit model. Now, the induced fit model basically says that the enzyme's active site has a certain shape that's somewhat complementary to a substrate, but that the shape gradually changes as the substrate enters into it to become more and more complementary to the substrate, and ultimately arriving at a perfect fit, so to speak. In other words, the enzyme's shape doesn't become completely complementary to its substrate until after the substrate is fully bound. Now here's a really cool example where we can see in red the enzyme hexokinase before it binds its substrate. After the substrate is bound, the enzyme's overall shape is shown by this ribbon colored in green. As you can see, the overall shape of the enzyme has changed somewhat during binding, as its active site is induced to fit more ideally around the reacting substrate. As you can well imagine, enzymes catalyze reactions by performing certain chemical transformations on their substrates once they're bound in their active sites. In order for me to be able to adequately explain how enzymes speed up chemical reactions inside living systems, though, I have to first teach you about amino acids. Now, I lament slightly that I would have already taught you this concept if I'd done our lectures in the order presented in the book. Due to schedule constraints, however, our Chapter 23 lecture on amino acids and proteins will have to wait for a later date. So now we digress. Amino acids, simply put, are small molecules that look like this. They're called amino acids because they have both an amine part, you see this NH2 amine part, and a carboxylic acid part. There are 20 universal amino acids found most prevalently in nature, and the only way in which each of those amino acids differs is in the identity of this side chain R. Amino acids generally bond together by having the amine from one amino acid condense onto the carboxylic acid group of another amino acid to form an amide bond, like the one shown here. Now, a chain or polymer of less than 50 amino acids is called a peptide. A chain of 50 or more amino acids is called a protein. So in essence, all proteins are really just made up of one or more large chains of amino acids. So just like a house can be formed by binding numerous bricks together in a specific fashion, a protein is comprised of numerous amino acids bound together in a specific fashion. Amino acids are the building blocks of proteins. Proteins' individual characteristics, of course, are caused by their having different numbers, lengths, and types of amino acids in their structures. Now, just so you know, all enzymes are proteins, but not all proteins are enzymes. You see, enzymes differ from other proteins in that they are proteins that are built specifically to catalyze chemical reactions. So yes, enzymes structurally are also made up of numerous amino acids bound together in one or more structurally specific chains. I'm now going to teach you about 3 different enzymes and how they work. I don't require you to memorize any of this. I just want you to pay attention to see how cool enzymes are, and then afterward be able to marvel at and appreciate the amazing chemical machinery that's all around and inside of us. As mentioned, a chain of 50 or fewer amino acids is called a peptide chain. Now in nature, and more specifically in our pancreases, as well as other places, I'm sure, there exists an enzyme called Carboxypeptidase A. When our bodies need to break down a peptide chain by chewing off one amino acid in that chain on the rightmost end, as depicted here. So in other words, if we've eaten a protein, our body needs to break it down by chewing off this end. It does so by employing this enzyme Carboxypeptidase A. This enzyme, in essence, catalyzes this overall reaction. It basically just cleaves the nitrogen-carbon amide bond right here to separate out the rightmost amino acid and puts in its place a negatively charged oxygen. This is the overall reaction. So the question is, how does this enzyme catalyze this reaction? Here's how. The peptide chain that's going to be shortened is ushered into the carboxypeptidase A's active site, as depicted here. So this depiction right here is basically just showing the squiggly line is the remainder of the peptide chain, which could be many, many amino acids long. They've only shown the specific atoms of interest here. So this right here is a terminal amino acid portion that's going to be removed, and this is the amide bond that's going to be clipped or chewed apart by this enzyme carboxypeptidase A. Now, being an enzyme, carboxypeptidase A is of course also made up of numerous amino acids itself. Inside the active site of carboxypeptidase A, here are some of the key players, the amino acids that form this cage around this peptide chain that's going to be digested. One of these amino acid residues is an arginine up here, which has 2 amino groups in it that hydrogen bond with the carboxylate end of the peptide chain that's going to be shortened. The amide carbonyl here is bound by this zinc ion inside the enzyme's active site. The enzyme's glutamate residue over here deprotonates a molecule of water, which thrusts this hydroxide anion into the carbonyl carbon, and then causes these electrons to go up onto this oxygen. At this stage, these electrons now go back down, and upon doing so, they break this carbon-nitrogen single bond, causing this nitrogen to be protonated by the proton that was originally taken from water by this glutamate. This effectively breaks this carbon-nitrogen bond and releases this terminal amino acid from the peptide chain. It also gives me this carboxylic acid group dangling off of the peptide chain. At this stage, the nitrogen or amine terminus here from this amino acid reaches over, deprotonates this carboxylic acid, and gives me this positively charged amino acid residue, which is then released back into solution. This amino acid peptide chain, which has once again been shortened now, is then also released back into solution. Now, as you can see, the amino acids framework here in this active site is set up so that each of these individual key groups around here acts like a player on an assembly line, performing its own role to achieve the task at hand. Now let's talk about another enzyme called chymotrypsin. This is a digestive enzyme that can catalytically cleave the amide bond of an amino acid from within the middle of a peptide chain. How? Now let me begin by showing you this figure so that we can get a better grasp of what I'm actually talking about. If you have a peptide chain, we have chymotrypsin in the digestive tracts that can once again effectively clip from the middle of the peptide chain a nitrogen carbonyl bond, this amide bond here. This is slightly different from the carboxypeptidase A's activity that we saw before, because this nitrogen carbonyl bond does not have to be on the end of the peptide chain. This can be right in the middle of it. Now I want to, of course, show you how this enzyme does that. What occurs is the amino acid is ushered into the enzyme's active site, which is shown here in this blue glob around here. As you can see, these squiggly lines over here to the left and right represent the rest of the peptide chain that's just dangling off. This particular amino acid or amide bond here in the center of this peptide chain is ushered into this enzyme's active site. At this stage, an aspartate residue here inside the enzyme reaches over and deprotonates this histidine residue. The electrons here in this nitrogen thrust down, and these electrons come over and grab a hydrogen off of this serine that's an amino acid stuck inside the active site of the enzyme. The serine then thrusts its electrons into this carbonyl carbon, and these electrons up onto the carbonyl oxygen. That takes us to this intermediate here. Now this carbonyl oxygen that's negatively charged, along with this oxygen here on the serine that's now bound covalently to this central carbon. This oxygen here is kind of held in place by the serine's amino group and this glycine's amino group. Kind of like a victim in one of the 72 different Saw movies. While this has been being held in place, these electrons now get thrust down and it breaks this carbon-nitrogen bond. This nitrogen gets protonated by removing the proton here that's stuck on this histidine moiety. Electrons go in here, electrons go back here to neutralize this nitrogen's positive charge. And that takes us to this stage here. Now you can see that what's effectively happened is the entire portion of the peptide that was here to the left is now gone and released. Now all we have to do is clip this bond in here and release the rightmost portion. What happens? Well, this histidine moiety's electrons on this nitrogen come down here, and these pi electrons steal a hydrogen from water that's been ushered in from the cell. Water then, these electrons now thrust into the carbonyl oxygen, this acting as a hydroxide ion, and these electrons go up onto this oxygen. This oxygen, once again, like a victim in the Saw movies, is bound by the serine amine group and this glycine amine group. This is held here temporarily, and then the electrons come down, and then they restore, or I should say break this carbon-oxygen bond to deprotonate the histidine and revert everything back in the enzyme to the state it was in when this whole process began. You can see that when that occurs, this portion of the amino acid has now been released. The carboxylic acid portion of the peptide chain is deprotonated by the amine from the other half of the peptide chain to give these two species. What's the overall effect? Well, chymotrypsin has taken a peptide chain and clipped an amide bond right in the center of it. It has done so at a very specific site, releasing the two halves back into solution. Let me teach you about one more enzyme, and then I'll let you go. This enzyme is called aldolase, and it catalytically converts fructose 1,6-bisphosphate, an intermediate in glycolysis, into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. I realize now that I've said that sentence out loud that you guys are probably thinking, what in the sweet, tangy crap does that mean, Mike? Well, glycolysis is the process by which our bodies, as well as those of many other living creatures on this planet, break down the glucose that we eat into energy. This process ultimately converts glucose, a 6-carbon long molecule, into 2 molecules of pyruvate that are each 3-carbon long molecules. Now, honestly, if we began studying in depth each of the catalytic steps of glycolysis, it would require a lot more time than we have today. We won't yet, but for those of you who go on to take biochemistry from me during a later semester, we will, I promise. So the underlying point is this. During glycolysis, our bodies break a 6-carbon molecule, glucose, into 2 3-carbon molecules of pyruvate. How does this happen? Well, once again, there are numerous steps, which I'm going to glaze over. For now, I want you to just believe me, or you can look it up later on your own if you're so freaking smart, that the first 3 steps of glycolysis convert glucose into this molecule right here, which is called fructose 1,6-bisphosphate. At this stage, this magical enzyme, aldolase, takes this 6-carbon molecule and breaks it apart into 2 3-carbon compounds. We can see that in this overall reaction. Aldolase takes the 6-carbon molecule, this bisphosphate, or fructose 1,6-bisphosphate, and breaks it into 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So how does that happen? Prepare to be amazed. So here we see fructose 1,6-bisphosphate sitting in aldolase's active site. At this stage, one of the enzyme's active site lysine residues reaches in and forms an imine with the carbonyl carbon here. So you can all see this imine. At this point, a negatively charged tyrosine, you see this tyrosine here, reaches in and removes this proton here. These electrons get thrust down here, closing like a trap door, and these electrons now flip over here to form a double bond, and these electrons get thrust up onto the nitrogen to neutralize its charge. That effectively breaks this carbon-carbon bond right here, which arrives at this intermediate. You'll see here that what has ultimately occurred is the enzyme has severed this 6-carbon molecule in half and released this purple-colored molecule, which is a 3-carbon molecule called glyceraldehyde 3-phosphate. Now a few more things are going to occur. At this stage, this nitrogen thrusts its electrons down in here, and these electrons come out and remove the proton that was just taken by this tyrosine moiety. That gives me this iminium species here. And then at this stage, water comes in and hydrolyzes that imine to return the enzyme to its initial state and releases this molecule, which is called dihydroxyacetone phosphate. Whew. Now these 2 molecules in purple at this stage are now released and taken through the rest of glycolysis, ultimately providing 2 molecules of pyruvate on the terminal end of the disassembly line. Now I know that you're probably feeling like you just had 11,000 baffling new concepts poured on top of you. I know that because I felt that way when I first started learning this stuff for myself. For now, I'm not going to require you, really, to know any of it, but I do demand that you appreciate it. I mean, look at this. Nature has these enzymes, assembled to catalyze these amazing chemical reactions in such elegant and specific ways. Enzymes really are like little assembly lines where each component, each amino acid in the enzyme's active site, acts like a person contributing individually to the end product. I think it's absolutely amazing. The wonder and awe that I feel when I study biochemistry is just wonderful and awful. In any event, this brings our lectures to a close. Thanks for hanging in there and listening to my Chapter 24 presentation on catalysis. Cat, lice, catalysis, grape, who, grape who. A fond farewell to you all. Until next time.